replication competent virus in virus-cell culture.
Cultured, Vincent. They cultured it.
There's an FC component to the antibodies that we think bind and neutralize.
All right, the next one, SARS-CoV-2 infection and persistence in the human body and brain at autopsy. So in this paper they looked at, autopsies on 44 patients who died, and they did PCR to look for viral genomes. And this paper this infection and persistence is wrong because they cannot say that by looking by PCR.
Overall, we're able to isolate SARS-CoV-2 from nasal placode, sinus turbinate, bronchus, lung, and also in the non-respiratory compartment, the choroid sclera of the eye, the jejunum, the mediastinal lymph node, the heart, and the adrenal gland. We sought to estimate SARS-CoV-2 RNA levels that were strongly predictive of replication competent virus in virus cell culture.
Cultured, Vincent. They cultured it.
We performed regression analyses with associated ROC curves and found that a ddPCR value of 758 copies per nanogram of RNA inputted into the assay predicted replication competent virus with a 76% sensitivity, 90% specificity, and an ROC AUC of 0.887. A subgenomic RNA value of 25,069 copies per microliter of RNA predicted replication competent virus with 72% sensitivity and 100% specificity with an ROC AUC of 0.915. These analyses provide a robust framework to predict a tissue run on ddPCR and or subgenomic RNA qPCR to determine if it is likely to have replication competent virus without having to perform cell culture in a BSL-3 setting, which is obviously a limited resource.
And look what they say here. We show that SARS-CoV-2 is widely distributed predominantly among patients who died with severe COVID. Okay, SARS-CoV-2 RNA, and that virus replication is present in multiple respiratory and non-respiratory tissues, including the brain early in infection. No, you cannot say that virus replication is present in multiple tissues, right, Amy?
Right.
By a PCR assay.
Right.
Come on, neurologists, or whatever you are.
They're not virus people.
They're not virus people. You and I are virus people.
Yes, I'm virus people.
Okay, the next one. COVID, this is from, where is this?
Nature.
Article? Nature. I think it's open access, so you can check it. SARS-CoV-2 infection and persistence in the human body and brain at autopsy. This is by a large collection of people all over the place. Looks like a lot of people at the NIH and other places as well. And, so the question is, does SARS-CoV-2 persist more than the 7 to 10 days than the acute infection? So acute virus.
Just to do authors real quick, Sidney Stein is the first author, Daniel Chertow is the last author, and there's the gazillion people listed, and also the NIH COVID-19 Autopsy Consortium, which I assume is a gazillion more people. So this was a very large collaborative effort.
And this has, one of the authors is Emmy DeWitt, who was on TWIV. She's at the Rocky Mountain Laboratories. I recognize that name. Vincent Munster, also there. He was also on TWIV several times. All right. When there are a lot of authors, I get, I just don't want to do it. Thank you.
No. We're not going to read all the authors. That's
So does the virus persist longer than the acute infection of 7 to 10 days or so? And so what they've done here is to carry out autopsies on 44 COVID-19 cases. These are all unvaccinated people. So it's April 2020 to March 2021. All unvaccinated individuals who died with COVID. So notice how they wrote that, with COVID. There's a big debate going on now, whether you die with or from, right?
Yes.
Are you admitted to a hospital with or for COVID? It's a very hard question to address, because when you go to a hospital now, you have to have a COVID test. And
I'm pleased to say, hospitals are starting to phase out the routine testing.
Yes, they are.
because this was early in the pandemic. It was the critical thing that we needed most. If you go back to our episodes then, it was, oh my gosh, why can't we do more testing? Because that's what we needed to try and contain spread. And now that this virus is endemic, testing for it is just generating noise. And you're getting, so anybody who's admitted to a hospital will get a rapid antigen test and it comes up positive. And well, that's not why their leg is broken, right? That's they're in the hospital for something else.
That goes down as a COVID admission, though.
Yes.
In many places. That's
yes.
Right.
If you look at one of the extended data figures for the paper, it's extended data figure 2, they have a table with what was listed as the cause of death for all of the patients.
Yes.
They only have COVID listed for one of them, but almost all of them were either sepsis or pneumonia or diffuse alveolar damage. And so I took that at least to mean that largely these patients were suffering from COVID infection and sequelae thereof.
Yeah.
They mentioned in one place that there was someone who had a seizure that they thought was unrelated to COVID. But from what I could tell, these people were all positive, and it seemed as though the disease that they died from was some sort of COVID-related pathology.
Yeah, and this is early in the pandemic when that was why people were going to the hospital.
All right.
What were the dates of this specifically? I didn't. This is
the
2021.
patients were April 2020 to March 2021.
Yeah. Okay.
So that's why they're all unvaccinated.
So then they then they looked for viral genomes by PCR. It's a very well done study, I think. They did things very extensively and very carefully.
Come on, neurologists, or whatever you
Well,
are.
They're not virus people.
And they do PCR. They look at virus, infectious virus. We'll talk about that. They also do genome sequencing. They do in situ hybridization. Right. So by PCR, they find they can see viral RNA in 84 distinct anatomical locations. Of course, the highest RNA level is in the respiratory tract, always, right? Because that's the main place where this is reproducing. They say, and this is a key point of the paper, they say the RNA is detected across multiple tissue groups among all late cases, so the ones that died longer after infection, despite being undetectable in plasma in any. It's also found in the central nervous system in 5 of 6 late cases, including one patient who died at day 230 after onset. So they still find viral RNA there. Now,
another important point I just want to bring up here is they standardize their procedures to the extent possible for these autopsies. So one of the issues when you're looking at postmortem data is, well, okay, how long did the body sit around? How was the autopsy done? Which tissues did you section and which didn't you? But in this case, because it's the NIH autopsy consortium, I guess they were able to come up with a standard procedure. And so these samples are actually comparable.
When they talk about the early versus mid versus late patients, that's based on something relative to the day of kind of illness onset.
Yeah.
But in every case, these patients are still ill.
right.
Yes.
Right. So previously, other groups have detected viral RNA in non-respiratory tissues. And they always say, well, a little bit gets in the blood and it spreads to other tissues. Doesn't mean it's actually reproducing there. And so they say, well, we don't find it in plasma. So that can't be an explanation for what we see. And I can't really argue with that. I mean, I would like to argue, but I can't argue. They don't find it.
Well, they also, we're going to get into, they go further than just PCR as well.
They also, so that those results are for genomic RNA. Then they look for subgenomic RNA, messenger RNAs. And they also find this as well. And that's more suggestive of reproduction, actual virus reproduction, because, the genome can just be there. But if it's being copied into mRNAs, it may be reproducing, more likely to be, I would say. So they find that those results are consistent with the genomic PCR as well. And then what they do is they take some of this tissue, which is ground up, and they put it onto vero cells and culture, right? And they say, do we isolate virus? And they can isolate virus from 25 out of 55 specimens, okay? Over a wide range of CT values or CQ values. But it's not quantified. This is one of my complaints about the paper, that I don't know how much virus is there, because if you put a piece of tissue in cells and there are a few PFU, it'll score as positive. And I don't know what a few PFU would mean in terms of pathogenesis. I would have loved to see a quantification of the tissue, but I understand that that's difficult. We used to do that all the time with mice, right? We'd infect them with polio, we'd grind up the brains and do plaque assays. So I'm used to it, but that's
You can't just grind up the whole human brain into a plaque assay. That's a little cumbersome.
Yeah.
So that's, I think, my main beef with this. Because as you see, they're going to conclude that we prove that there's extensive extra respiratory reproduction of the virus, and I'm not sure we can say that, but
Do you think the tissues were too autolyzed for EM work?
Did they do some? I think they did some EMs later on, if I remember.
Because if they were finding whole particles, then that's signs that it's
Does
reproducing.
anyone remember?
I only rember the in situ and sort of the histo.
Yeah, no,
path.
I'm thinking
I
of the
don't
other
think
paper.
they did.
Yeah.
Yeah, the other papers. Yeah.
There's
The other
lots of
paper. So
EM
no
there.
EM. I don't know. Maybe it was, not suitable
far
for.
gone.
I just don't know.
Yeah.
But if I saw one particle, I still wouldn't say
No.
right, extensive reproduction. They did some viral genome sequencing, and they see some synonymous substitutions. In other words, changes that still code for the same amino acid outside of the respiratory tract, which they say is additional evidence that there's reproduction, right? Because it's different from the virus in the
Yeah,
respiratory
yeah,
tract. Okay. And then they do in situ hybridization, where they're looking for, in this case, spike mRNA in tissues. You take a tissue section and you hybridize it with a nucleic acid probe, which will show you the presence of viral nucleic acid inside of cells. And they have a very nice image showing this, where the RNA will show up as a color, and you can see it's inside cells, right? So I think they said how many of these? 35 different cell types. How many people did they do this in situ with? And I don't see the number popping. It's 16 cases covering a 4-log range of N gene copies. So they do see this, and the pictures are pretty. You can see the cells, nice morphology, and you can see viral RNA in them. I don't know if that means there's active replication or not.
What would active replication look like?
Well, one of the things they note is that there is no cell lysis or inflammation in any of these sites.
Yeah, but this is dead tissue, though. Come on.
Well, it's dead tissue now, but it was recently alive. And
It's a good point.
if the virus was in there replicating, and this is a lytic virus, you might expect to see some cell lysis.
Yeah, Brianne, would you expect inflammation to go away immediately after death?
I don't think that you would. No, you can certainly see inflammation in other tissues in other disease states by autopsy. I wondered more about, whether or not, for obvious reasons, I was thinking about, what's going on with sensors in those cells to be sensing viruses and things like that. And I didn't end up taking a look at some of the data on that I had planned to look at. But you certainly could see inflammation.
Yeah. So that may suggest that there, maybe there's RNA around, maybe there's a little bit of infectious virus, but it may not be reproducing extensively. I mean, we know now for RNA viruses that they can persist for a long period of time. Diane Griffin was one of the first people to show this, that we think these are viruses that come and go, but actually the RNA sticks around a long time.
Yeah, and I also think that these figures are particularly important because of that idea that had been out there previously that this is only because of blood being in some of those tissues that the virus happens to be there. We can see that it's actually in the cell types of the tissue.
Yeah.
I guess
So
it's
I
kind of
agree.
odd that,
Go ahead.
sorry,
No,
that
go
we
ahead.
see this, like in figure 2, you can see that it's obviously inside of like many of these cells, extrapulmonary tissues, and that there isn't any inflammation is kind of odd, I find.
Yeah, that's why I'm sort of wondering, particularly when I'm thinking about the brain, how much sensing really there is, and how much we would see inflammation in other cases as well. I think it may be kind of a immune privilege dampening of not having a lot of inflammation in some cases in those tissues.
What does the word exemplar mean? The best example of?
A really good
A
example.
good
of.
example of.
An example that you hold up.
Okay, so this
An
in
example that
situ.
exemplifies.
So they say these are exemplars. So maybe these are the best sections they saw. Maybe they're just a few in every tissue, spotty patches of infected cells, and that's it.
And they're really diverse tissues too. They have spleen, adrenal gland, endometrium, testes, esophagus. Like they're not, like they're very, very diverse tissues.
yeah.
And the fact that there's we shouldn't say replication, I guess, but we can see RNA inside of the cells and that there's no inflammation whatsoever. At least in these, maybe there was in other sections. I wonder that they're.
So they're still looking for a cause of death. Is that right?
Well, the cause of death of these people has already been stated on the death certificate, right?
Yeah.
And as
They
Brianne
didn't
said,
die
these
for
are
a cytokine storm or anything. like
right.
that,
So these
obviously.
these are causes of death that I think, with only one exception, are completely consistent with dying of COVID-19. It's pneumonia. It's the usual things. And there's one kid who had a genetic seizure condition and had a seizure and died, and that's okay. So that kid died probably of the seizure, but everybody else seems to have died of COVID-19 or related complications.
I had to look up, they listed a lot of the causes of death as DAD. And so I had to look up what DAD was as a cause of death. It was diffuse alveolar damage.
Oh, no. So anoxia, you think they died from low oxygen levels in the body?
Well, yeah, if the
Perhaps.
I mean, you have to nail it down to some physiological event.
Right.
Yeah.
DAD is listed for many of them.
Okay.
Mm.
Okay.
Dad killed him.
Right.
So that's pretty much it. They also see RNA in the brain, but again, they see no histopathology anywhere.
I maybe didn't say it clear enough. When we first tested to try to grow virus in the brain, we failed. Like, in other words, in cell culture, we failed. We used a standard virocell line. The sequencing results that I showed you show evidence of minor variants in the brain, and sometimes not minor variants. Actually, they're nearing equal distribution of variants with non-synonymous mutations that we detected in the brain that we did not detect in the respiratory tissue, suggesting that ongoing viral replication does take place in the brain and that there actually is some evolution of the virus in the brain. We have tried to grow virus from the brain. We're not done. We're going to persist. So far, we have not definitively accomplished that, but it is something that we continue to pursue. But when we found by our sequencing that there were viral variants in our brain samples that were not present in our lung samples, we said, oh, we got to look again. And so by that time, there had become a new reagent, and the new reagent were this modified viral cell line that express the human ACE2 and the TMPRSS2 receptor, that cell line, although I can't say definitively, but there's a suggestion that cell line is more permissive. So we tried again, and we in fact did grow virus out of the brain. We grew virus out of the brain, and we confirmed that the virus that we grew out of the brain was indeed the same sequence of the virus that we had detected from our RNA later preserved.
So they say these findings conclusively demonstrate that the virus is capable of infecting and replicating within many tissues, including the brain. And it just brings me to mind, we had a neurologist from Columbia on 2 years ago, and they had done a similar study at autopsy and found no evidence for
Right.
virus. Jim Goldman was
Yeah,
involved in
yeah,
that study, and I forgot her name. It's escaping me, but
Kieran
Thacker. Thank you. Kieran Thacker. She was the neurologist who was on that paper, and she came in and did the TWIV. So she said, I remember asking her, is there any evidence for reproduction? She said, no, we don't have any evidence. So I don't know why there would be a difference in the two studies, but obviously we need more work to sort it out.
Well, the
Now-
closer you look, the more you see. So no evidence for viral reproduction means you didn't see it. And these folks saw it. They also did a very, very detailed search for this exact thing. So it may just be, it was there, but it wasn't visible with the available assays,
So I think-
the assays that were used.
Yeah, there's some infectious virus present, but it may be low. We don't have a quantification, and there's no inflammation. There's no cell damage. There's no histopathology. So I don't know what relevance to the disease it has.
Ladies and gentlemen, we got him.
Okay, because I know these individuals would like to think this has something to do with long COVID, right?
Sure.
But I don't see any evidence that it's contributing to extra respiratory symptomatology. I mean, I agree that in the nasal epithelium, there's evidence for, destruction of the support cells and also lymphocyte infiltration that may be persistent. That's fine. But here, I don't see convincing evidence that any of this would be playing a role in disease, I guess.
A really difficult, expensive study that I would love to see, that would be hard to get done, I think, is to do this for some other RNA respiratory virus. Do it for,
Yes.
pick a maybe rhinovirus, maybe whatever.
Influenza, where people
influenza,
die, right?
Sure.
Yeah.
Actually, flu would be a great choice because you could get a bunch of people who definitely died of flu or of flu-related complications,
Right.
and go search as deeply as this with these same procedures and see if you find flu, because long flu is not really a thing as far as we know, but if you get similar results, maybe this is just something that RNA respiratory viruses do.
Right. And I think the other piece, and again, this would be such a hard study to do, I don't know if you could totally do it, would be, these patients all died of COVID. Even the ones who were sort of long or the late time points were really dying of COVID related pathology. And so what you'd want to know if you were sort of tying this to long COVID is, can you still see this after the patients have recovered?
Yeah.
And it's not clear that any of these patients had recovered.
Right.
And we could do the same thing in Alan's cohort as well, just making an even more impossible experiment.
Yes.
Yeah.
I wonder if, because
I
they
like
don't know.
that. Go ahead. Sorry.
Sorry, they don't know. I guess it doesn't say if this was, they didn't sequence the actual virus, did they? Because we don't know if it's the
Yeah.
ancestral strain or if it was one of
Yeah,
the
they did.
variants. Or if,
They did sequence.
They did
so is
genome
this,
sequence.
but I mean, they did genome sequencing, but for every single patient?
No, for
So do we know?
a subset. Yeah.
Mm-hmm.
1,
'Cause I
2,
wonder-
and 3. It looks like they did. Wait, we used genome sequencing to look at spike gene only in a total of 46 tissues in 6 individuals. Just the spike, though, not the whole genome. Yeah.
Yeah, I wonder if depending on the variant, because obviously at that point in time, I'm not sure which variants would have been circulating already, if that would influence the tropism of the virus itself.
Yeah, they actually say that. This was before the emergence of variants, so we don't know how that would affect the tropism. Yeah. I do think that they say that, these are all from old people who were unvaccinated and died.
Well, except
And-
for the kid.
Except for the kid, they had pre-existing medical conditions. So who knows whether the same would happen in a young, healthy, or a vaccinated person, right? They may not.
And with the variants, yeah, the things have moved on since this, so we don't know that the current infections look like this.
And they say, although it is tempting to attribute clinical findings observed in PASC to viral persistence, our study was not designed to address this question. Yet you bring it up.
Because it was tempting.
It was tempting.
Well, and you also know that the readers/press might bring it up.
It has not escaped our notice that
Yes.
this could, yes.
James Watson.
This is a Nature
Francis
paper.
Crick. Yeah, I mean, obviously that's in the back of everyone's mind, but I'm not convinced that because of all these limitations that this has broad. And I think looking at another virus would be great because I suspect you'd find it with other respiratory viruses as well.
If you look at the literature, for example, influenza, I don't think that folks have taken the type of approach that we have to characterize influenza viral distribution across the body and brain. But having said that, people have been studying influenza for a really, really long time and have done a lot of careful studies. And those studies do suggest that the prevailing knowledge and dogma is that those viruses, at least the predominance of them, absent the high path avian influenza viruses that have this special polybasic cleavage site that allow them to get outside of the respiratory tract, that the seasonal influenza viruses that we're infected with don't do this.
It's really exciting because we can get about maybe 40 times more sensitivity, which may be required if we're looking for very, very low amounts of persistent virus. If SARS-CoV-2 is persisting in tissues or making proteins in tissues, or even potentially replicating in tissues, then it may be in very low levels. This is not going to be a very high level system. This is going to be analogous to HIV, where people on therapy will have very, very low levels, but that low level can still set off an inflammatory cascade that can have clinical consequences. So although it's low level, doesn't mean it's not insignificant. I think that's important to note. And so the Explorer scan will hopefully help us to have even higher sensitivity to identify those areas where this low level viral production is within the body. So it's super exciting. And our colleagues at UC Davis have been really very collaborative in kind of sharing their technology for some of these seminal studies.
That's great. And then you're doing the same thing with T cells in long COVID too, right? So you're actually you can take it. This is what's extra cool about what you're doing. You can, as long as you can tag something with a radio particle that the PET scanner can image, right? You can choose different, as long as Henry on your team or your team members can do that, you can actually also make a what ends up being called a radioligand for T cell activity too. And you've done that, right?
That's right. We have already done that, and we've actually already imaged many people with and without long COVID symptoms, and we're analyzing that data now, that initial batch. So we're very excited to see what we'll find. But yeah, and we've been doing this with HIV as well, and that what we're using this instead of being a monoclonal antibody, which is specific for a viral protein, for example. This is a small molecule that is selectively taken up and retained by activated T cells. These are both CD8 and CD4 T cells. And T cells are a part of the immune system that's intricately involved and extremely important in antiviral immunity. So they will traffic and become activated in areas of active viral infection, for example. Other things such as inflammation as well, but they are one of our main antiviral adaptive immune responses. And so what we've done is we can use this small molecule called FRAG, that's made by Cellsite, a company here in San Francisco. And when we inject this into the vein, this actually has a fluorine, radioactive fluorine, and not a zirconium. So the half-life is very short. It's just a few hours before it disappears. And so what we do is we inject it in about 45 minutes or an hour or so after injecting this. We then do this PET CT. And this allows us not to look at viral proteins per se now, but what it does is it allows us to see where these activated T cells, the antiviral kind of immune response is located. So for example, is there an increase in T cell activity or trafficking to various tissues? Could be gut, could be the brain. This crosses the blood-brain barrier easily, so we can see if there's any immune response happening in the central nervous system. We shouldn't have a lot of T cells in the central nervous system normally, so if we see those, then that means that there's something drawing those cells into that space. And so this is really exciting because not only can we have the capacity with the immunoPET imaging to look at the virus itself, but we can actually look at immune responses that may be responding to the viral infection or viral persistence as well. So we're using this two-pronged approach where we could look at virus directly, but we can also look at the immune response to a potential virus infection, or even an aberrant immune response without virus. So for example, if there's autoreactive immunity or autoimmunity that develops, that we would potentially also see that with this T cell imaging agent. And so this has been super exciting because we can now use this to look essentially across the entire body in real time to see where a potential aberrant or kind of overzealous immune response may be going, which is one of the thoughts that could also be leading to a lot of the symptoms that people are experiencing after COVID-19.
Absolutely. And do you try to the greatest capacity to have the same study participant do the immunoPET with the tagged monoclonal antibody for the SARS-CoV-2 spike protein and the imaging that tags the molecule that can find activated T cells? Because it seems really cool if you could overlay that and say, look, this person had viral SARS-CoV-2 reservoir here, 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 and they had T cell activity here, here, and here too. And you could basically almost overlay that understanding.
Absolutely. So the goal going forward is to image these different modalities in the same patients or the same participants, you know, the same people. Yeah, absolutely. And so you know, this FRG, this activated T cell marker, only stays in the system for a few hours before the radioactivity decays and it's excreted pretty quickly. And so it's gone pretty quick. So we can image folks, and then any period of time point later, as closer probably the better, we can then do the immunoPET imaging with the radio-labeled monoclonal antibody, and we can get a sense of is there a coordination between these T cell responses and potential viral persistence, or is there a disconnect? Right. You know, are T cells not going to where we're seeing a persistent virus? Would suggest that it's not just an overzealous response, it's a misdirected response, and we're actually not getting to the compartment that we need to get to. So we don't know which one that is, but hopefully that these types of studies will inform, you know, let us know, you know, is this just an overzealous response to ongoing infection, or is it an aberrant, dysregulated, ineffective response to viral persistence over time? So these are the types of information that we can get from these studies.
Very important information. And then I know that you're also set up to actually collect tissue and gut or lymph tissue from even potentially the same study participant that underwent the monoclonal imaging and the T cell imaging, to actually verify via the biopsy if you also might find SARS-CoV-2 spike in the tissue biopsy itself, right? There's almost just yet another form of validation.
Absolutely. So we've been working with our kind of clinical director of the late cohort, Dr. Michael Peluso, in our group, and what we've been doing is that we've been collecting, and we'll continue to do so going forward, of course, tissues after we've imaged. So we don't want to do it directly after imaging because we don't want radioactive tissues to be long. But if we wait a week or two, it's okay. And so what we do is a few weeks after we complete imaging, we will do either a sigmoidoscopy or a colonoscopy to get colorectal tissue or potentially distal ileum, part of the small bowel as well. And we take multiple pieces from the bowel, and then we can do everything from viral detection using PCR for RNA, or we can look at in situ tissue looking for proteins, and we can also look at host genetic responses and to see how those correlate with persistence of potential virus or viral parts of the virus within that tissue. And so that's ongoing. And then we can correlate that with what we're seeing on the PET imaging. So for example, the T cell imaging agent or the immunopet monoclonal antibody to look for viral persistence. And so we'll have a complete picture. We're also now moving forward with lymph node biopsies as well, where we take cells from the lymph nodes, and then we can also do the same types of studies that we do in the gut tissue to see if we see viral persistence, and if we do or we don't, how does that correlate with some of the PET imaging, and how does that correlate with the host immune responses within those tissues directly as well. So we've done this within our HIV cohort. It was very telling that, you know, in folks where we saw increase of the HIV-specific antibody tracer, we actually saw increase in cellular HIV protein, for example, in those participants. And so this is the type of work that we're now moving on to in the setting of SARS-CoV-2 that really give us a whole picture, and also to verify that we know what we're seeing on the PET scan is what we're looking for. You know, there's always a potential that there could be non-specific signal, or it could be binding to something else, or maybe it's getting trapped in a compartment for another reason. Maybe the antibody that we give that's supposed to bind to the virus actually makes an immune complex with something that's circulating and is deposited into a tissue. And so we really want to figure out what we're seeing with that PET scanning. So what we can do is, by looking directly in the tissue, although we're just looking at a tiny fraction, remember, of the entire body, whereas the PET scan can look at everything, we can at least correlate those two and say, okay, in the region where we did the biopsy, what did we see on those PET scans? And that's very important.
That is very important. That's huge. I'm not sure if I've ever seen a group that's as well set up to study tissue, just what's happening in tissue, infection, immune response as your group. It's usually the opposite, which is, we know most teams, which is okay, but most teams draw blood because it's the easiest, well, whatever, it's commonly ethically approved, like to get ethics approval to draw blood is straightforward in this, and we capture what we can from blood. But I think one of the things that's difficult when it comes to persistence of pathogens is that with a lot of viruses, studies have shown that over time, their genetic material that tend to clear from the blood over time. It's not really the smartest place to be, and they go into tissue where they hide, sort of in simple terms. So if we don't look in tissue, you could easily come up with the wrong conclusion and say this person doesn't have the virus anymore, when really it's just not in their blood, but it still could be in any other location. And that's what your research helps to address, which is really important. Just to clarify, if we could clone you and your team, you could.
Well, that'd be scary.
It'd be scary, but maybe worth it. It depends. Maybe worth the risk. You could technically, depending on what you could iterate, you could tag, let's say, a therapeutic or monoclonal that could also maybe find another, like Epstein-Barr virus protein, or a cytomegalovirus protein, or a Borrelia Lyme pathogen, I don't know, antibiotic like you could actually do this and potentially image deep reservoirs of many pathogens. Would that be reasonable? Is that true?
Absolutely.
Yeah.
Okay. No, this is just the tip of the iceberg. I think although, you know, our immediate expertise are retrovirus infection, HIV, and now SARS-CoV-2, clearly, you know, we have interest. You know, for example, human herpes viruses, we know are persistent chronic infections that have a latent, but also a lytic or active form, and they can flip in and out of those depending on, you know, environmental stress or other factors. And we and other groups have also showed that there might be an association between reactivation of these human herpes viruses and the development of long COVID over time. We're still trying to piece together what that is or how that's doing it, but there certainly is this significant association between evidence of EBV, or Epstein-Barr virus, reactivation and long COVID. Now, you know, 95% of the adult population in the US will carry EBV, and some mostly in a latent form for most people, but and so it's very possible that those folks, or at least a subset of people that kind of have this acute inflammatory kind of response to COVID-19, which is an incredibly inflammatory and immune dysregulating process, could be reactivating, and that could also be kind of adding fuel onto the fire, I guess you could say, in terms of kind of, you know, revving up an aberrant and dysfunctional immune response and inflammation that could kind of have longer term impact, even regardless of viral persistence of SARS-CoV-2. And so, but not only that, but, you know, I mean, these human herpes viruses, EBV and CMV, most particularly, but other ones, that can cause Kaposi sarcoma, for example, or other things are of huge interest because, they reactivate in folks that are immunosuppressed, whether it's, solid organ transplant or treatment for rheumatologic disease or stem cell transplant and, long immunosuppressive use. And they can have, terrible clinical outcomes. People can get very sick from reactivation of some of these. In fact, with EBV, there's tumors and cancer that have been associated with reactivation, as well as many other, potential autoimmune processes. And now there's a potential link with multiple sclerosis or MS with EBV infection. And so I think that these are clearly the next low-hanging fruit, that we want to look for. Where is it? How active is this virus by looking for viral proteins that are produced within different tissues, within different situations? So absolutely, this is something of definite interest of us. If we had more bandwidth and funds, and we have antibodies that have been clinically made in a grade that's acceptable for human use. This is the big challenge with getting into people, right, is we need antibodies that are produced in what we call a good medical practice, kind of a way of that the FDA will allow us to give it to people. And it can be very expensive to make these. And so we've relied on either commercial antibodies or antibodies that have already been made for human use in other studies. But, certainly going forward, this is something that we would be extremely interested in developing and applying the tools that we have developed already for these different viruses. And bacteria as well, as you brought up. There's a group I know here at UCSF that's interested in imaging TB and persistence as well. And so, it's not just limited to viruses, but any pathogen that can be recognized with a monoclonal antibody, you can do immunoPET imaging.